Hello, I'm Dr. Liam Crowley, and I'm a postdoctoral researcher at the University of Oxford. Today, I'm going to update you on current efforts to sequence the genomes of the UK Aculeata. In this talk, I'll introduce the Darwin Tree of Life project and explain some of the aims and reasons behind this project. Then I'll give a general kind of introduction and summary of what genome sequencing is, before moving on to talk about the specifics of the Whiteham Genome Project. And then to finish, I'll just show some of my personal highlights from the last couple of years of sampling effort. So the Darwin Tree of Life project is kind of a successor to the Human Genome Project, which took just over 12 years and more than $4 billion to sequence a single genome. That's the human genome. The Tree of Life project wants to do sequencing, but rather than doing one species, we want to sequence every single species of eukaryotic organism in the UK. So very ambitious project. It's a very interdisciplinary and collaborative project. So you see here just some of the key partners that we have involved with the project, and it's funded by the Wellcome Trust. The aims of the project is to produce full genomes, so that's chromosomally complete, telomere to telomere, so all of the genetic material of a species, as well as a significant portion of these being annotated, so the actual genes labeled on those genomes. This allows the genomes to be used by a wide variety of different researchers. We want to do this for every species of eukaryote in the UK eventually. So that's over 70,000 species from the most recent lists. However, the project breaks down into 3 phases. So we're currently in phase 1. So from around 3 to 5 years worth of effort, we are aiming to sequence 1 or 2 species from every single taxonomic family in the UK. So it's about 2,000 families. Obviously, there'll be some families which we just won't get because they're scarcely seen or they're obscure or they haven't been recorded for many decades in the UK. Whereas there are other families which we will target in much more depth, many different species from that family. And these will be families of particular evolutionary or ecological interest. Phases 2 and 3 then seek to ramp up and scale up the sequencing efforts to eventually sequence every species. But why are we trying to sequence all of these different species? Well, first of all, this is the first time in all of human history where this is actually even possible, that we could actually do it. And this is largely because of the development of new and exciting sequencing technologies, such as long-read sequencing. So as you can see from this graph here, the cost of sequencing has rapidly dropped over recent years. This is actually a log scale axis on the left. So that gives you a sense of just how quickly the prices have dropped from very, very expensive to actually now a price which is feasible to do many thousands of species. But just because we can do something isn't always a good reason for why we should do it. So there are other reasons why actually doing this massive sequencing effort is really useful. First of all, there's a huge potential for discovery of, for example, new compounds, which could be used in medicine, or other things which could help us from the fields of engineering to economics. And in a way, we don't know what we don't know. So there's lots of exciting potential there. The other thing is, by studying genomes, we get a much better understanding of evolutionary biology. And so we can look at how genes evolve and how they interact with each other, and shed new light on the different relationships between species and genes as they have evolved in different species. And finally, and arguably most importantly, genomes are fast becoming an essential toolkit for 21st century biology, whereby they are useful to people outside of the field of genetics and genomics. From ecology to physiology to behavior, having an understanding of the underlying genomics behind an organism is really transformative in what we can learn about different species, and potential for conservation and protecting species and during unprecedented ecological decline that we're currently facing. So what is genome sequencing? Well, in brief, it is just working out the complete set of genetic material that an organism has. So the genetic material is stored as DNA, this molecule, which is organized in as long sequences of nucleotides, so A's, T's, C's, and G's. And these are arranged and organized into chromosomes. So when we talk about sequencing the genome, we are reading the genetic material in these chromosomes and also other genetic material which is held outside of chromosomes, for example, mitochondrial DNA, and determining the order of the A's, T's, C's, and G's in this sequence. So it's a bit like reading a book and translating a book. This can then tell us about how genes and the DNA itself is organized. Now, the catch is that there's an awful lot of genetic material inside each and every cell. And in fact, these genomes are enormous. So in arthropods alone, they range from about 320 million base pairs in some very simplified genomes of plant parasitic mites, all the way up to being over 13 billion nucleotide bases in grasshoppers and crickets. So once we've worked out the genome for an individual, and for the Tree of Life project, we are doing this from a single individual, that produces a reference genome. So in much the same way that a type specimen is the reference for the morphology of a species concept, these reference genomes are in a way a genomic type for the genome of that species. Then this can allow us to do lots of other things in the future. For example, once you have a reference genome, you can find particular regions of the genome and use that to then resequence. So you can go out and get lots of samples from many different individuals, and then much more easily and cheaply, you can then just sequence a specific part of the genome, and you can do things like working out related lists, which is really important and useful in conservation genetics, as just one example. Now, at this point, I'd just like to highlight the difference between genome sequencing and DNA barcoding. So DNA barcoding actually relies on a very specific part of the genome, which is actually only around 600 base pairs long. So that's roughly 0.00003% of the entire genome. Now, similarly to how a barcode in a supermarket works, the idea is that every species has a unique combination of this region, which is different from other species, but consistent within a species. So these regions have to be chosen very carefully so that they have enough difference between different species, but there's not very much variation within a species. So for most animals, we use the cytochrome oxidase 1, which is actually a mitochondrial gene involved in respiration. So it's very important, and without it, the organism can't survive. So because it's so important, it's quite highly conserved, yet there's still enough variation between different species of animals that we can tell just by looking at these 600 bases. However, if you want to take a barcode and work out what the species is, you need to have a good reference library of barcodes, and there are projects ongoing trying to populate a complete barcode reference library for all of life. So very exciting times in terms of species monitoring. So on the Whiteham Genome Project, as I said, this is part of the Darwin Tree of Life project based at Whiteham Woods and the University of Oxford. And this is, in a sense, a genomic observatory. So because we have the context of the long-running historical experiments at Whiteham, we're trying to put these genomes within the context of the ecosystem, and we're trying to sequence as many different species from this ecosystem as we can. But our focus is mostly on arthropods, and within that, mostly on insects to date. So Whiteham Woods is around about 1,000-acre site just to the west of Oxford, owned by the university, and it has a range of different habitats. So we have good extensive areas of ancient woodland, but we also have other stands of different tree species and different ages. As well as the woodland, we also have large areas of calcareous grassland, actually quite high quality calcareous grassland, as well as various different habitats, including wetland habitats such as the fen and several large mature ponds. So good opportunity to find lots of different species. So before we can actually collect a specimen for genome sequencing, we have to build up a list and have an idea of our targets. What species are we going after? What species are we likely to find, especially at Whiteham when we have a limited site? Once we know roughly what we want to find, we can then go out and collect. And this is always taking live specimens, which can then be taken back to our field lab facilities. Once we have a specimen, it's very important then we have an accurate species level identification. So there are various different ways that we can get to this. The majority of the specimens, at least so far in the project, have been based on field identifications. So things like this nice Dolichovespula media, where we can be very sure that we know the species based on characters that we can see from a live specimen in a pot or in a net. However, we are, as you'll be aware, this is not feasible for a huge amount of the UK species list. So we've been working on other techniques that we can use to accurately identify live material, because we need the specimens to be live when we process them to preserve large, long fragments of DNA. So one of the things we've been working on is knockdown. So this is mostly actually using refrigeration. So we put the samples into the fridge for about 10 minutes. Cool them down, and we can then put them under a stereo microscope and see the characters we need to see to identify them. We've also potentially going to use CO2 knockdown as well, although so far the fridge knockdowns have worked pretty well for most taxa. Something else which we can do is, if we know there's a specific piece of morphology, we can actually dissect that off as we process the specimen, and then the rest of the specimen can go for genome sequencing, and then the bit of morphology we can use to identify. So a good example of that would be the genitalia. If we have a male and we can easily get the genitalia out, then that's a good way to accurately identify. Another method we've used, and this is only used for a very small number of specimens, but this is using conspecifics. So if we have collected multiple individuals and we have a very high degree of certainty that they are the same species, for example, they're siblings, then we can take one to voucher and identify traditionally, and then the other one can be sent off for genome sequencing. And finally, again, barcoding has only been used for a very small number of specimens, whereby we remove a leg, send that off for DNA barcoding, compare against the library. If that species is in the reference library, then we can say with a high degree of certainty it is that species, and then the rest of the specimen goes for genome sequencing. However, that relies on it being in the library, and every specimen that we are sequencing we are also barcoding, but that's as a confirmation of identification, not as an identification itself, because we need to be careful about doing that. Once we have a specimen, we know what it is, we can then process it to prepare it for DNA extraction and genome sequencing. So we're using long read gene sequencing, which relies on large fragments of DNA. So in the same way that a jigsaw with a few large pieces is a lot easier than a jigsaw with lots and lots of small pieces, we want as large fragments of DNA as possible. Now, as soon as an organism dies, the DNA starts to break down into smaller fragments. So the best method to preserve the DNA is to actually snap freeze it, so rapidly cool it to around -80. So we do this using dry ice or liquid nitrogen. As the specimen is freezing, we can dissect larger specimens like this Dolichovespula here to use manageable chunks of tissue. But for many species, because they are small, they are just processed whole. And then we can also remove a piece of tissue, normally a leg, for taxa which have legs, for barcoding. As I said before, this is confirmatory barcoding, not identification barcoding. So in just over 2 years of sampling at Whiteham Woods, I've collected just under 3,000 samples, which come from just under 2,000 individuals. And this represents 1,287 different species. These are mostly insects with a few other arthropods and other invertebrates as well. And within the insect is mostly the most speciose orders, so Hymenoptera, Diptera, Coleoptera, Lepidoptera, and Hemiptera. And so far we've done just over 250 families. In terms of the Aculeata specifically, we've got quite a good range of families covered. Now, there are some families we are still missing. This is either because they are not found and not going to be found on the site, or they are obscure or scarcely seen, or just difficult to identify groups. So this is something that I am working on, and I'm hoping to pick up a couple more families in the next year. But the main progress in the future will be trying to flesh out and get as many different genera and species as I can. But because Whiteham is quite a good site, it's quite large, lots of different habitats in the middle of kind of southern England, it's got quite a good list, and we've got quite a good representation of that list so far. So just to finish with some of the highlights from my sampling, the first highlight would be Osmium bicolor, not because it's a particularly scarce species, but because Whiteham is such a fantastic site for it. On the right kind of day in the spring, when it's warm, you will see absolutely dozens of individuals flying around. You can watch them provisioning in snail shells. It's really fantastic site for this species. Another species of bee, which I was really pleased to find, I found a really good population of Mcropus europea down on the fen. So to the kind of southeast of the site, there's a really good fen with lots of yellow loosestrife, and yeah, sure enough, the bee turned up in good numbers as well. And then we were really pleased to find Andrena hatta on the site. There's a good areas of kind of calcareous grasslandy meadows with lots of scabious. And last year we found Andrena hatta on the site. It seemed to have a really good year last year, actually. It turned up at lots of new sites in Oxfordshire and other counties. Another Andrena, which I was pleased to see, again, not because it's particularly scarce, but because I found good numbers of them across the site, was Andrena bucephala. And in particular, I found communal nests, which was nice to see, and which also allowed me to find another highlight, which was Nomada hirtipes, which we found in and around the communal nests of Andrena bucephala. So that was a good bee to add to the site list. Another new bee for the site list was Svacodesniger. This was found last year. I found a couple of males flying around an area which was known nesting location for Lasioglossum moreo. Unlike the other highlights, this species actually hasn't gone for genome sequencing, because it wasn't till later when I identified the specimen which I realised what it was. I wasn't able to get a field ID on this one. So hopefully I can pick this up again in the future from a live specimen, and that can go for genome sequencing. And likewise for the only wasp which makes the list. Unfortunately, it hasn't been amazing for wasps, but I recently had Chrysoceros congenna at the site. This map is almost certainly out of date. I think the species was new to Britain in 99 and seems to be expanding since then, like many species. So I'm sure there's lots of other localities that this is turning up now across the south and central England, including now Whiteham. So that just leaves me to acknowledge all the people who are involved with the project. So I have here a list of the people who've been involved with collecting and identifying samples on the Whiteham Genome Project from various different taxa. Most of the Aculeates have been identified by myself and Stephen. I would also like to acknowledge all the people working on the project kind of downstream. So we've been doing a lot of sample acquisition, but without the people doing barcoding, compliance, extraction, sequencing, assembly, creation, publication, we wouldn't end up with that final publicly released full genome sequence that is available for researchers around the world to use. So yeah, massive thanks for them and their efforts on this project. And thank you for your attention listening today, and I'd like to invite any questions.